We live in a very interesting time with regard to the creation of large-scale biobanks. Large-scale biobanks will be very important for pulling out certain questions in genetics and very important for pulling out certain questions about disease and about its pathology. My hope is that large-scale biobanks will ultimately be the way that we can really begin to nail down questions of prognosis. From the ENIGMA perspective, large-scale biobanks offer both an interesting challenge and an interesting opportunity. The challenge is that you'll have single data sets that will have very large amounts of information, very large numbers of large amounts of genetic data, large amounts of imaging data. And at one level, they seem to be competitors of what ENIGMA is. But I don't really see it that way. I think actually it's the opportunity. That is, that ENIGMA has worked through different ways of generating phenotypes, has done a really important job, particularly of late, of working on new methodologies that you can extract important information from. And I think being able to apply ENIGMA-related protocols or things that were developed for ENIGMA in these large-scale databases will be incredibly important. It's not simply creating one large biobank that's important, but it's the methodologies that you can use, the robustness of the methodologies that you can use in order to be able to have robust and consistent phenotypes that will be as important as creating the single repository. ENIGMA has been very successful in writing certain kinds of papers to date. Those papers have helped us understand some of the genetic architecture of brain structure. They've also helped us understand differences between cases and controls in different disease groups. But as ENIGMA matures, we have much better possibilities of helping the scientific community. And that will happen in two ways. One will be by us presenting and publishing null results to say, look, we didn't find this in the largest samples that exist. And by being careful about it, by showing what the differences were by group looking at these effects. Because I think for the field to move forward, we need to stop always asking the same exact questions. Another way that ENIGMA, as it matures, has a way of really helping the field is through the development of new modeling and methods technology. And in the cases where those work, that's fantastic and we can present that. But it's also just as important to present when they didn't work. If we're utilizing different types of multivariate models to try to predict who is in a disease group and who isn't, and we can't show an effect across sites, that is really important for the field to know, because in science we get very infatuated with a certain type of model or a certain type of effect. But if we can't show it in a large group setting like this, then its utility overall becomes much less robust and much less stable, and we have to be very cautious about it moving forward. ENIGMA has both an ability and a responsibility to begin to publish papers with null results, and to publish papers where we've tried methods that have failed. In those ways, I think ENIGMA not only will continue to grow, but will continue and become an even more important aspect and resource in the field.